Hello everyone, thank you for joining us today. My name is Sinan Yavuz and I'm a bioinformatics scientist here at Seven Bridges. We have an exciting presentation planned for you today with our friends from eNIOS who have developed the BioInfoMiner app on the Seven Bridges platform. Throughout the webinar, if you have any questions, you may ask those through the Questions tab that is visible on the right-hand side of your webinar interface. But before turning over the mic to Aristotle, I'd like to give a brief overview of the Seven Bridges for those of you that might not be familiar with us. Founded in 2009, we employ one of the largest groups of bioinformaticians dedicated to genomic analyses. Over the years, we have been honored to have been named a trusted partner of the NIH, one of the smartest companies in the world by MIT, and a multi-time winner of the BioIT Best of Show Award. Seven Bridges offers a complete bioinformatics ecosystem, enabling researchers to discover like never before. Our unified analytic platform, the Seven Bridges platform, serves as the core bioinformatics foundation, enabling dispersed teams to collaborate in a secure, compliant, and efficient manner. Our platforms currently offer the only multi-cloud functionality, enabling researchers to process data where it resides, reducing the data logistics burden, therefore accelerating the rate of discovery. Within the Seven Bridges platform, researchers have easy access to public and proprietary datasets to create, view, or run analyses and explore the raw data and results. In addition, there are more than 400 pre-built tools and workflows across a wide range of research areas that are ready to use. But not only do you have access to these pre-built tools and workflows, you can also port your own workflows into the platform and automate them for standardized high-volume processing, all while maintaining the data integrity through extensive tracking and data provenance, ensuring the long-term reproducibility of your analyses. Here, I would like to also mention a specific product we are very excited about, which is Graph. Graph-based pangenome references offer the possibility of increased accuracy in detecting insertions and deletions, as well as longer structural variants by alleviating the reference bias, ultimately improving precision, speed, and recall without compromising specificity in the variant calling process. This technology, the Seven Bridges Graph Genome Suite, was recently published in Nature Genetics. In addition, rounding out our bioinformatics ecosystem is our expert scientific services. Our on-demand professional and consultative services are custom fit to meet your needs. Whether you're looking to optimize existing workflows, require assistance with complex workflow development, or even seeking scientific peers to help determine the best analysis to perform, we are here with you every step of the way. By leveraging the breadth of the Seven Bridges bioinformatics ecosystem, researchers can go beyond standard workflow processing to one of through iterative analysis. Now you can analyze genomic and phenotypic data in a secure and collaborative cloud environment that helps you refine analytic workflows for improved accuracy, computational speed, and efficiency. Once established, every group of scientists in your organization, including bench scientists, can use automated workflows to process massive amounts of data for secondary and tertiary analyses. Again, all while remaining centrally within the Seven Bridges platform. By harnessing these core capabilities, researchers are able to enhance hypothesis resolution processes, identify actionable biomarkers of interest, and even turn failing clinical trials around. Over the years, one area of primary focus of Seven Bridges and our clients has been centered on identifying actionable biomarkers, which is why we have partnered with eNIOS in 2018. I would like to now introduce Professor Aristotle Hatzigiannou, the CEO of eNIOS, to talk about this partnership and their application called BioInfoMiner on the Seven Bridges platform.
So, good afternoon. Thanks so much, Sinan, for the kind words. We're really proud to be part of the Seven Bridges ecosystem. The reasons are obvious, because this environment really provide us all these means in order to catapult our development effort and come up with much more efficient solutions in the area of genomic interpretation, which is our target area. So in this webinar, we're going to examine the importance of interpretation regarding the processing of genomic data. We will try to explain how it differentiates from other processing steps. We will review the critical challenges regarding robust interpretation, and we will try to see how computational intelligence may optimize the interpretation tasks in an industry-ready fashion. So, active players in the genomic analysis business are pretty much aware of the significant progress that has been made recently, either in the digitization of the readout of the experimental techniques that are available in order to record and accumulate results from various types of experiments, or in the analytic platforms which crunch these data streams that are generated by such experiments. However, the image is not the same for biological interpretation, which is currently still manual, slow, and lacks standardization. So one reason for that is that data are way more complex than what is expected. And in order to shed light regarding the contribution of distinct bioinformatic processing tasks in the whole analytic pipeline, we review this typical workflow that summarizes the typical steps of bioinformatic analysis and interpretation. So the first 3 stages describe different processing steps, which have to do with the quality control, the trimming, with the assembly, the signal estimation, the generation of bulk target lists using different types of statistical tests. And all these steps represent the first type of analysis. However, if we want to make meaningful associations and gain biological insight, which means concrete pathways, networks, functions, or much more to evaluate and trace the dynamic behavior of whole systems, and further on to try and derive meaningful biomarker sets, which would allow targeting of reliable targeting of drugs, robust cohort stratification, and improved predictive capacity regarding diagnostic, prognostic, or response to therapy purposes. These 2 steps refer to interpretation. Speaking about interpretation, it actually boils down, it boils down to the extraction of actionable information from large genomic bulks. This really means scalable prioritization of few important molecular processes and pathways, which should ideally be largely orthogonal in terms that they describe distinct cellular processes. It also has to do with the retrieval or inference of the underlying network topology. And last but not least, the extraction of ordered sets of regulatory biomarkers according to the interconnection with these pivotal molecular processes. So the experience of the various NGS types of data that we are increasingly accumulate show that the interpretation is far more complex than what would be expected, and this is partly attributed to the very, very large covariance existing in biological data. A strong message and a caveat regarding these high throughput techniques, which are really amass this data, and for instance, GWAS techniques are similar techniques, is that despite the fact that they may be powerful in monitoring thousands of features, they're not perfect enough to deconvolute noise omnipresent in these measurements. So as a result, we are coming up with thousands of putative targets, a large part of which are really false positives and highly fallacious. So it is important to bear in mind that statistical correlation does not mean causal association, and therefore is different from interpretation. Robust interpretation would require modeling based on biological concepts rather than mere statistical correlations. So, reviewing the state of the art regarding biological interpretation, we would say that it is characterized by lack of consensus methodology. Tasks are therefore performed manually, heavily relying on experts' prior knowledge. The applications that are built are normally designed as expert support tools. They're not data-driven, which means that they're not dynamic enough. So that means that when you enter a different input, you do not get that different, similarly different output. They are very, very sensitive to small changes of parameterization. And all these point out that there is huge bias towards pre-recorded statistical correlations and pre-recorded biological knowledge. Last but not least, the broader community knowledge is severely underexploited because the type of statistical tests that are used are simplistic enrichment statistics. In order to remedy this problem and break through the limitation that we just described, we really try to exploit technologies from the field of ontological processing. And that means that we try to exploit corpora of functional terms that have been developed by different scientific communities. Such corpora are the gene ontology, the human phenotype ontology, the reactome pathways, for instance, KEGG pathways. And all these corpora follow a very specific structure, a three-type structure of their annotations, which is actually something very, very interesting from the computing perspective. However, these ontological annotations are sparse, biased, fuzzy, and inconsistent. Their analysis is based on simple enrichment statistics, and therefore we derive thousands of tedious and highly redundant terms. And this type of results do not provide a scaffold for biomarker discovery and prioritization per se. In order to resolve all these problems, BioInfoMiner implements an innovative algorithm for information extraction and modeling, which is applied on ontological graphs that have been designed after we have automatically corrected these community annotations. The key features of the BioInfoMiner approach is firstly the intelligent semantic processing, which transforms bulk annotations to consistent knowledge networks. Then omic data, which are coming out as the resulting data from a specific experiment, are projected to these knowledge networks that have been delineated. And then artificial intelligence methods, data mining and statistics, are applied to the semantic space because network properties are much more conserved than individual molecular features are, which tend to follow a much more erratic pattern of manifestation from the perspective of personalization. That means that different persons are exposing different vulnerabilities in different genes, but these genes tend to converge and perturb the same instrumental molecular networks. Finally, the network-aided noise reduction, which corrects the annotation bias, promotes and unearths systemic processes and driver genes. And these systemic processes are key cellular processes which are functionally largely orthogonal. That means that they are highly distinct and they incorporate different set of genes. So we feel that the best analog to describe biological complexity is that of the iceberg. Biological information extraction, which means that in order to find which are the causal cues behind a phenotype that we are studying, is more or less like tracing a needle within a haystack. And even worse, the big data mean that everything is big, and that means that noise too is very, very big, is much bigger. So traditional statistical approaches emphasizing in the significant lists of targets which represent the limited perception of the phenotype that we study. However, what we would like to see is to unearth these underlying complex mechanisms and hidden relations which are below the surface of the sea. But the problem then is at the same time that we should rule out the technical noise, because the models that we would otherwise build wouldn't have the good generalization ability due to the overfitting. Another big problem is that even if we use the genes of the significant list, in this part of the list, we have always strong presence of noise, which means that we have high false discovery rates. Therefore, we really need to forge another approach, and this is the network-based modeling of biological knowledge and exploiting the ontological processing and trying to sort out information in a way that it will bring to the surface systemic processes. How BioInfoMiner makes that happen and mitigates this burden? Firstly, sorting systemic processes. The sorting of the systemic processes does not take into account only the statistical enrichment score, but takes into account the position of these terms which have these high enrichments to the semantic graph. And this is feasible through the application of intelligent semantic processing techniques. Having done that, we are able to highlight critical ontological terms which map to gene cliques that would describe gene subsets with highly concerted function, more or less like a Gog-Will system. Then the critical hubs from the regulatory point of view would be genes standing among these different gene cliques, which enable a systemic spillover. And in order that we are able to trace these important hubs, BioInfoMiner implements a unique combination of ontologies which represent a very good scaffold for genome-wide searching with PPI interaction networks, which provide high quality information about the interactions of specific gene products in the vicinity of these important hub genes. So, reviewing the different software that has been developed in the area of interpretation, we would categorize that to the three academic software, which is fairly far from being data-driven, as it is not standardized, it's not optimized, it's extremely sensitive to parameterization and to user settings. And it might be suitable for basic research, but it doesn't have generalization ability, and it cannot run in an iterative fashion. Regarding the current commercial solutions, these are still highly exploratory, they are overly descriptive, they are relying on supervised learning, and they mostly support the decision made by experts, and those two are not data-driven. On the other hand, BioInfoMiner derives concise, robust, reproducible results in an automated fashion. These results are not sensitive to small changes in parameterization. It delivers intelligent information processing. The results that it generates are, as we said, highly automated and standardized, so they are industry ready. And in this way, BioInfoMiner generates results suitable for streamlined, large-scale, and supervised applications. BioInfoMiner generates a series of results in a highly structured output format. So the first part of these results has to do with the derivation of the pivotal pathways, where the ontological terms that are pinpointed dictate the delineation of hierarchical semantic graphs. These strong correlated and semantically clustered terms shape systemic processes. And in order to infer in the other step the master regulatory genes, these genes are linked with these systemic processes which seem to be more remote functionally when we are reviewing the physiology of a cell. Regarding the gene part of the signatures, first of all, these signatures are highly compact. And as a result, we are coming with highly informative heat maps, as the one that you see here, which links systemic processes with master regulatory genes. These signatures possess a very high information to size ratio. The prioritized genes have very strong association with the physiology that we study. So even mild changes might have large impact on the physiology that we study. And the interpretation that we perform about the physiological state that we are interrogating is being done at the level of the signatures rather than that of the single genes. And in that sense, this interpretation is much more robust. In the end, this interpretation is not relying solely to the quantitative results that are related with these genes, but actually to it exploits the knowledge about existing interaction of these genes within them and with the other genes of the input list. And in this sense, it represents a biologically inspired feature selection method. Applying BioInfoMiner to other types of processing workflows that have been built by other groups, it can really provide value to the results of other solutions. And here we present, for instance, the use of BioInfoMiner in order to further prioritize a list of important variants. So we know that when we perform whole exome sequencing experiments, we are filtering out variants. Normally, we might have variants in the order of 1 million, and then when we see the impact of the variants on the protein function, we are coming down to the order of the hundreds of thousands. If we emphasize on rare variants, we might come down to the dimension of some thousands. And if we try to filter out even more, we would go to several hundreds of variants with known clinical significance. When we apply the BioInfoMiner network prioritization methodology, we can come to the point of a few dozens of important variants according to their network effect. And now I'm going to present you a very, very powerful result and feature of the BioInfoMiner application, which is the pharmacogenomic analysis. At the moment, BioInfoMiner supports two modes of pharmacogenomic operation. The one has to do with relating the genes of the signature one by one with reports about potential drug targets that can be used in order to exert an effect on these specific genes. And in this way, what you see here is the network of the gene signature of hub genes describing an initial schizophrenia signature. And you can see that the hub genes are highly connected, and actually they describe different aspects of the schizophrenia. Some of them, they have to do with the anti-inflammatory component, which is present in schizophrenia because it's a neuroinflammatory, it has a strong neuroinflammatory component, but also there are drugs which highlight the known antipsychotic activity that should be administered in order to improve the therapeutics effects of schizophrenia. The other mode of operation is the one that actually exploits the whole signature, and we make use of the L1000 connectivity map in order to highlight which of the part of the signature that we have derived can be associated with the expression that certain perturbagens, that is compounds, chemical or drug compounds, have exerted on specific cellular models and have been recorded in the L1000 database. So in that sense, we are able to trace out a subset of genes which have the same expression profile as the genes of our signature, subsets of genes with opposing profile to that of our signature, and of course we can detect the overall common perturbed genes, not taking into account the direction of the expression. As a BioInfoMiner application is part of the 7 Bridges platform, it can support and is nicely integrated with the other tools of the 7 Bridges platform. Therefore, we have already tested and developed workflows integrating BioInfoMiner seamlessly to workflows provided by the 7 Bridges platform. And we believe that this is actually one of the big assets of the 7 Bridges platform, because it provides very, very strong, in terms of efficiency, high fidelity workflows that really gave us the opportunity to catapult our efficiency in delivering high-end solutions in the area of interpretation, where we would like to emphasize as a company. So at the moment, we support solutions regarding whole exome sequencing, RNA-seq, whole genome bisulfite sequencing, but in the near future, we will extend that in order to support whole genome sequencing analysis, taking into account either microRNA analysis or long non-coding RNA analysis. The way that we do that is that we seamlessly integrate the BioInfoMiner application with the workflows provided by 7 Bridges, and in that way we can really automate the whole processing starting from the raw or the processed data that are related with a specific experiment, in order to come up with a small, highly compact molecular signature, which can, of course, provide the means either for drug repositioning or precision medicine or diagnostics or prognostic studies, as we have already showed you, and as we will show you also in the case study that we will present in a few minutes. So speaking about the case study, we have really used a TCGA, head and neck squamous cell carcinoma, that we have access through the 7 Bridges platform, and we were able within minutes to extract a highly meaningful signature Here you can see the workflow that has been already developed, and it is part of the 7 Bridges platform, which links the different tools of the 7 Bridges platform with the BioInfoMiner signature extraction module, which provides 3 types of results. One that has to do with the visualization of the results, the analytical module, which provides the results for the gene signature analysis, and the extended analysis modules, which provides the results for the pharmacogenomic analysis. And in this way, we were able, out from an initial input signature of 939 significant genes related with the head and neck squamous cell carcinoma dataset, we were able to infer a highly compact signature of 23 genes when we use the gene ontology, which describes the hub genes regarding the manifestation of head and neck squamous cell carcinoma when they were compared with their adjacent tissue controls. It was even more interesting the fact that we were able to stratify the samples by characterizing the samples according to the expression they had with in the genes of the signature that we first derived. So the samples that had low correlation in the expression with the genes of our signature had a much better, a statistically significant improvement in their survival, according to the samples that had high correlation with the expression of the genes of the signature. And we further tested that with random signatures of the same size concerning the significant genes that had been used in order to infer the hub gene signature. So at this moment, I will switch to a short live demonstration. Sorry. So what you see here are the results of the analysis that has been run on BioInfoMiner regarding the head and neck squamous cell carcinoma dataset. And actually, we should go here, where we can see different implementations of this signature. So I will pick this one. And here you can see all the outputs. I should have to say here that in order that you run this analysis, you really need to come and register to our platform, the BioInfoMiner platform, so that you can get a token that you can use for your analytic purposes. And then you can either use the eNIOS division of the 7 Bridges platform, or your own division if you are customers of 7 Bridges, in order to integrate the BioInfoMiner analytical capabilities to your workflows. So, for instance, the type of results that you would get regarding the head and neck squamous cell carcinoma is this graphic synopsis. Here you can see an ordered list of hub genes. The red color in the gene cycle describes the direction of expression. Red is for overexpression, blue for underexpression. If you hover the mouse over a certain gene, you would see all the agent pathways that this gene is linking. The different colors describe different eigenpathways. And as you can see, as we are hovering and we are moving the mouse towards the right, we can see that we have less and less eigenpathways being evoked. This is the type of the semantic graph display that you get. There are also other options. There is the bar type display where we have an ordered list of all the significant processes. Results are displayed not according to the functional terms, but actually according to the systemic processes. These are a cluster of terms. And in this way, we can see which are the genes that are related with these significant pathways and processes. And the third type of display is the informative heap map linking semantic processes with the hub genes. And actually, by hovering around over the descriptions of the systemic processes or the terms, you can see which terms are clustered to which systemic process. And we also integrate and take into account the expression of these genes. And actually, we can see that seemingly different physiological processes, like the abnormal pulmonary alveolus epithelial cell morphology and the abnormal hormone level, are actually very much related in terms of genomic content. And these type of insights are the kind of highly innovative insights that uniquely BioInfoMiner can deliver. So I will switch back to the presentation. Sorry. So, sorry for the technical hitch. As a summary, we would say that BioInfoMiner is a powerful application for automated, actionable interpretation of omic data streams. The key features about BioInfoMiner is that it is dynamic, data-driven. It delivers dynamic, data-driven information extraction. BioInfoMiner results are also network-oriented through the intelligent fusion of semantic networks and PPI networks, and they manage to accomplish huge dimensionality reduction. It is important to take in mind that BioInfoMiner delivers highly compact, robust, and informative omic signatures, and the compactness is a key feature which opens the way for a wealth of application with clinical interest. Because if we don't have this compactness, it is impossible to target in a specific fashion with pharmacological compounds specific genes. The spillover could be huge, and the problems would be due to the non-linearity of the physiology of the cell would be immense. So BioInfoMiner empowers numerous interesting applications in the field of pharmacogenomic analysis regarding epidemiological profiling and stratification and integrative omics. And in the near future, we plan to release new modules which will enable comparative analysis of single cell sequencing data and interpretation of metagenomics data. So should you like to learn more about BioInfoMiner, please find us at eNIOS.com or bioinfominer.com, or follow us on Twitter at eNIOS or at Facebook. Thank you so much.
Thank you very much, Aristotle. I just want to remind our attendees that you can ask your questions through the Questions tab in the right-hand side of your webinar interface. With that said, we have a few questions already came in. Aristotle, these are for you. The first one would be, what are the main differences between BioInfoMiner and the other similar software?
Yes. First of all, I would say that BioInfoMiner is the only tool to provide a combination between semantic ontological vocabularies and PPI networks. These are vocabularies and functional descriptions which are coming from two different scientific worlds, and overall they provide very, very complementary and highly valuable information. To the best of our knowledge, there are not systematic solutions that integrate this type of information in a standardized context, which means that it should be unsupervised and highly automated. Moreover, BioInfoMiner delivers an analysis which actually performs semantic clustering in these corrected annotation graphs, because the initial annotation graphs that we derive from ontologies is, as we said, highly fuzzy, highly redundant, and highly prone to trivialism, which means that if we do not perform correction, the type of conclusions that we will get will always tend to highlight already extracted knowledge. So for me, these are some very, very strong differences that BioInfoMiner has with respect to other solutions, and which make it unique. One last could be that it is inherently data-driven, which means that when you give a different input, you get a different output. But this data-driven nature of BioInfoMiner is the result of the first two things that I told you: the unique combination between ontologies and PPI networks, and the fact that we perform this type of intelligent semantic clustering on the graph of the corrected annotations.
Thank you. We have another question that is asking, what does the integration process of BioInfoMiner look like?
The integration in terms of information integration, we can really exploit different vocabularies which have the same type of structure, so this tree structure, which means that we can actually come up using the same type of standard methodology with results which provide new insights, simply because different communities have studied different things. And this is evident when you compare results that you get from one ontology. People tend to use overly the gene ontology, but there are other ontologies which are more interesting, like the human phenotype ontology, which is an ontology created by which is a very appropriate ontology for the study of genetic syndromes, and which is actually highly valuable if we are to dissect the contribution of each gene in complex pathologies. Or there are other ontologies like the mammalian phenotype ontology, which has been developed using model animals, mice model animals, and which provides a very, very accurate functional annotation of the diversity of phenotypes that describe numerous diseases. Also, BioInfoMiner, we could say that is able to provide the same type of analysis in completely different streams of data, because it reviews the role of a gene at the functional level. So that means that we can readily integrate a transcriptomic chunk of data, a proteomic one, a methylation one, and so on. So in that sense, is a very, very powerful platform for integrative analysis. And I already mentioned that we plan to have a module for comparative analysis of single cell sequence data. The challenge here is that if we wouldn't use network persistent motifs and we would use individual molecular targets, then we would have the problem of the lack of, let's say, of a common basis. Whereas by reviewing and tracing out the important networks, we are able to associate the pathologies or the phenotypes that we study with instrumental molecular processes. And this is, to my understanding, the only rational way in order to compare data sets of a extremely high dimension, as single cell sequencing data are.
Thank you, Aristotle. We have another question, which is, can the user export the visualizations produced by BioInfoMiner in image files?
Yes, of course. This is a characteristic that has already been developed and supported in our pipeline. It is something that it is not, unfortunately, at the moment ready for the 7 Bridges platform, but of course we give access to all these, tab delimited TXT files or the HTML files that can be used as basis in order to reproduce the data. And as I already told, if one would like to use BioInfoMiner, he would have to register at the moment in our platform too. So and that means that he could have also direct access and download results from there. It is something that we, however, plan to integrate in the next months, also to the 7 Bridges platform.
Thank you. I have one more question in the list. You have touched upon in your answer to the first one, but maybe it's useful to expand on a bit. Mm-hmm. In what way BioInfoMiner is data-driven?
Yes, as I already told you, a very, very simple way to understand whether software is data-driven is that to rate its efficiency to respond to different cues with different results. So that means that if we use as input a certain list, or if we compare, or if we use different input lists, we should expect to a certain extent different results. Of course, we should always bear in mind that these lists should describe different phenotypes. That being said, that means that the hub genes would be different. And this is something that we have persistently tested, examined, and we are actually very, very confident that we do it very, very well with BioInfoMiner. And we do that well because we perform a multi-stage correction at the level of regarding the quality of the annotations that we exploit. The first versions of these annotations that are incorporated in ontological vocabularies are pretty fuzzy. They are pretty vague in what they describe functionally, and as a result, they're characterized by high redundancy, and they tend, if you do not correct them for the bias that relates certain genes with certain functionalities, they tend to pinpoint the same already well-known targets. But by performing this correction, we are able to come up with novel insights. And actually, there are many publications that have been made with BioInfoMiner, which were not just top quality publications, but they have also made it to create new patented products.
Great. Thank you very much, Aristotle. With that said, we have exhausted all the questions from our attendees. So I would like to thank everyone for joining us today. And for any other information, you may reach out to eNIOS at the details displayed on the screen, and you can reach out to Seven Bridges team via sevenbridges.com/contact. And we will also send out the recording of this webinar after the completion of the webinar. Again, thank you very much for joining us today, and hope to see you in other Seven Bridges webinars.
Bye-bye.